Welcome back to my YouTube channel Learning Biophysics with Team 320. Science fans, today we are diving into the fascinating world of DNA replication, the process that ensures life keeps on going. DNA, short for deoxyribonucleic acid, holds the genetic instruction for building and maintaining an organism. Think of it like a blueprint, a detailed set of instructions for creating you. Every time a cell divides, it needs to make a copy of its DNA. This ensures that each new cell gets a complete set of instructions. Without DNA replication, a life as we know it would not exist. It's fundamental for growth, repair, and the continuation of living things. Now let's break down this essential process into bite-sized pieces, starting with the first step, initiation. Picture DNA as a twisted ladder. The famous double helix. To replicate this ladder needs to be unzipped. This unzipping happens at specific points called origins of replication. An enzyme called helicase sweeps in, breaking the weak hydrogen bonds holding the two DNA strands together. This creates a replication fork, a Y-shaped region where the DNA strands are separated. Ready for the next step, like elongation. But hold on, we are not done with the initiation yet. Before we can start building new DNA strands, we need a primer, a short piece of RNA that acts as a starting point for DNA polymerase. Think of it like this: you can't build a house without foundation, and you can't build a DNA without a primer. Now let's move on to the exciting part, building the new DNA strands. With the DNA unzipped and the primer in place, it's time for DNA polymerase, the star of the show, to shine. DNA polymerase is an essential enzyme that adds nucleotides, the building blocks of DNA, to the growing DNA strand. But here's the catch: DNA polymerase can only add nucleotides in one direction, from the 5' end to the 3' end. This means that one strand, called the leading strand, is synthesized continuously, while the other strand, the lagging strand, is synthesized in short fragments called Okazaki fragments. It's like building a Lego tower with one hand constantly adding brick while the other hands add them in chunks. It's a bit more complex, but you get the idea. Now let's see how this replication party comes to an end. Replication complication. Replication completion. The termination after DNA polymerase has done its job. We have two identical DNA molecules, each consisting of one old strand and one new strand. This is called semi-conservative replication. Pretty cool, right? But how does the replication process stop? Well, it varies between different organisms. In prokaryotes, which are simple cell organisms, replications ends at a specific termination sites. In eukaryotes, which are more complex organisms like us, it's a bit more complicated. Eukaryotic chromosomes are linear and replicating the ends, called telomeres. It is a bit tricky. This where telomerase comes in, an enzyme that helps maintain the length of telomere, but that's a story for another time. Now, the essential toolkits. Enzyme involved in the DNA replication. DNA replication is an team effort. And like any good team, it needs the right tool for the job. We have already made helicase and DNA polymerase, but there are few other key players we need to introduce. Primase. This enzyme creates the RNA primers that DNA polymerase needs to get started. Ligase. This enzyme acts like a glue gun, sealing the gaps between Okazaki fragment on the lagging strand. Endonucleases. These enzymes are like molecular scissors cutting DNA at specific point. Single-stranded binding proteins. These proteins bind to the separated DNA strands, preventing them from re-annealing and keeping them stable for replication. Together, these enzymes work in perfect harmony to ensure accurate and efficient DNA replication. It's a beautiful example of molecular teamwork. Now, next section will be our prokaryotic versus eukaryotic, a tale of two replicons. While the basic principles of DNA replication are universal, there are some key differences between prokaryotics and eukaryotics. Prokaryotics, being simpler organisms, have a single circular chromosome and single origin of replication. Eukaryotes, on the other hand, have multiple linear chromosomes and multiple origins of replication per chromosome. This allows for faster replication, which is essential for this more complex organism. Furthermore, eukaryotic DNA is packaged into chromatin, a complex of DNA and proteins, which adds another layer of complexity to the replication process. But despite these differences, the fundamental goal of DNA replication remains the same: to faithfully duplicate the genetic material and ensure the continuity of life.